Welcome back, Life Science learners. In our segment now, we wrap up our revision on nutrition. Let's get straight into the lesson and try and attempt some questions based on deeper understanding around processes like the regulation of blood sugar levels. I have a question for us to work on. Let's get straight into that and try and analyze how questions based on the regulation of blood sugar levels can be assessed in an assessment. So the question reads, the graph below shows the results of a glucose tolerance test on a healthy individual, person A, and on a diabetic person B. After fasting for 10 hours, they each were given a drink of glucose solution containing 50 grams of glucose. The amount of glucose in their blood was then measured every 30 minutes for the next 3 hours. So guys, it's important to have some context to a question like this. Essentially, it's important to understand that sometimes individuals that go in for a glucose tolerance test have to undergo a period of fasting. And that means essentially that the previous night would have been the last meal that they would have had. They would have slept through the night, got up the next morning, and then gone into a clinic or a facility that would have tested their blood glucose level. And that's essentially important to be able to establish what the baseline glucose level is. Notice that these two individuals, person A being a person that is not affected with diabetes, and person B, a person affected with diabetes, were then both given a fixed or a known amount of glucose solution, and that then was monitored over the next 3 hours in terms of how that level would have be affected in their blood. So let's look at the graph as we unpack this question. So the graph then illustrates the results of the glucose tolerance test of the healthy individual, person A, and a diabetic individual. Let's look at the key. So the healthy individual is indicated with triangles, so that's the healthy individual here. If we just outline that there. If we're looking at person B is the diabetic, that's that line, and it's indicated with squares, so that's the individual there. Okay, and we have one more line which basically shows you a solid line with a circle indicating the normal blood glucose level, which is seen at this height there. Guys, this is measured over a period of time from minutes 0 to 210, so that's over a period of time. And on the y-axis, the blood glucose levels are measured in milligrams per deciliter. So that is essentially the stats or the information that was collected regularly every 30 minutes over a period of time between persons A and B, and then that is then compared to the normal blood glucose level, which we see as the baseline value. So guys, clearly we can see from this graph there is a significant difference in the level of blood glucose between person A, who is healthy, and person B, who is a person that is suffering from diabetes or is affected by diabetes. Let's move on to this question. What was the greatest concentration of glucose in the diabetic's blood? Again, guys, the question is very pointed to the diabetic individual. So if we have to look at that, so it's going to be graph which indicates the ones with the squares. If we look at the greatest concentration was this point here, and so we've got to use the information and with your rulers extrapolate that onto the y-axis, and in this case it is 300. Guys, the answer is not complete yet. It's important that we're able to include the unit that is given on the y-axis. So in this case it is 300 milligrams per deciliter. It might not make sense to you, the unit, but it's important that you provide a unit for your answer, and that is only when your answer can be accepted to be correct. So in this case, it's a simple extrapolation of data from the graph. Let's move on to a few more questions on this. From the graph again, determine how long it would take for the glucose concentration of the following. A, the healthy person to return to the level when the glucose solution was consumed. So in the healthy person, how long does it take for the glucose levels to return to the normal when the individual consumed it. So if you look at the initial amount was just under 100 milligrams, and it obviously reached that value after a specific time period. We've basically got to look at how long did it take for it to get back to normal. And this was approximately, if you plot that down, 150 minutes. So that took the individual 150 minutes to bring down the blood sugar levels back to what it initially was. Let's move on to B. The diabetic person to return to the level when the glucose solution was consumed. So when the individual had consumed the glucose, it was 200 milligrams, and if you look along the graph, this was when it returned back to normal. Again, you've got to determine how long did it take from that point. Again, if you extrapolate that down to the x-axis, you can see that it took that individual 210 minutes. So guys, clearly we can see that between both these individuals, this individual, which is the healthy individual, took much less time to bring the blood sugar levels back to what it originally was before or at the time when they consumed the sugar. However, the individual that is diabetic took much longer, and this essentially points to the struggles that a diabetic individual will have in maintaining their blood sugar levels and bringing them back to a normal level. An interesting difference that we needed to note. Let's move on to question 3. What effect would injecting insulin into the diabetic person have on the results of the test? So guys, remember that an individual that is diabetic has a problem with, again, being able to absorb the blood glucose into the cells and using that. However, we do know that insulin as a hormone will assist the individual to be able to allow the insulin to open up the cells so that the glucose can then move through from the blood vessels into the cells. So what effect would injecting insulin into the diabetic person have on the results of this test? It will reduce the amount of blood glucose in the blood vessels, therefore decreasing the levels of glucose in the bloodstream. Right. Question 4. What is the function of insulin? Guys, insulin is a hormone produced by the pancreas. Insulin, again, is released into the bloodstream, and as it travels into the bloodstream, it is able to allow the cells to absorb the glucose, therefore decreasing the glucose in the blood vessels, allowing for the cells to absorb the glucose so that it can be used for cellular respiration. So that would be the function of insulin. It allows for the blood to be absorbed, for the glucose to be absorbed into the cells, therefore decreasing the blood glucose levels. Often learners explain that the function of insulin is to decrease the blood glucose level. That is correct. However, you need to understand that it's the insulin that allows for the glucose to be absorbed into the cells, which then ultimately reduces the level of glucose in the bloodstream. Right. Explain briefly why insulin, which is a protein, is injected into a diabetic person rather than given orally. And this is a 2-mark question. And I'm going to pause at that point because there's a lot of information that's given to us around insulin. So guys, insulin is a hormone. And this specific hormone, again, is protein-based. Remember that insulin is injected into the bloodstream, and that then travels into the bloodstream to the cells which allow it to absorb the glucose. The question points, rather than giving it to an individual orally, it is injected. What is the reason for that? So guys, think about the pathway of insulin as it moves from the mouth. It will need to move from the mouth down the esophagus into the stomach. Being a protein, we know that in the stomach, protein digestion happens. So guys, if you orally take insulin, that insulin is going to go into the stomach, and being a protein, the insulin will be digested, hence making the insulin ineffective. Remember that the insulin then needs to be absorbed, and the absorption only happens in the small intestine. So it means that effectively insulin will be digested as a protein in the stomach and will lose its effectiveness and not be able to assist the cells in absorbing the glucose into the bloodstream. And that's essentially why it is injected directly into the bloodstream, so that it can go into the bloodstream and then effectively allow cells to absorb the glucose, therefore reducing the blood glucose level. So that was an interesting question. Let's get back into right at the start when we discussed the adaptations of the jaw and the teeth of different consumers for their mode of nutrition. So this question essentially points to that. Let's read through this. Study skull A and B below and answer the questions that follow. So guys, if we stand back and reflect, here we see skull A, which clearly indicates a large canine. You can see clearly large canines in them. We're seeing very well developed cheekbones. We can see triangular shaped teeth on the upper jaw and the lower jaw. This is significant in us understanding the mode of nutrition. And this particular skull will be that of a predator. How do I know that? I can see that you've got large canines. Let's look at skull B. Again, you can see that well developed and significantly elongated jaw, showing the presence of flattened molars and premolars. We see the absence of canines. So we see the absence of canines, but significantly well developed incisors. This would be significant in an organism that is a herbivore, one that consumes plant material. And so you can see that the incisors help them to bite off leaves and grass, and the molars and premolars, which are well developed for chewing and grinding of that plant diet. And that's essentially what you should be able to identify when comparing the skulls of different consumers. So let's move on to a few questions based on this. Identify which skull A or B belongs to a herbivore. As I mentioned, herbivores are individuals that will lack canines. They have large incisors. When we compare the jaw structure of both, we can see that these individuals lack canines, but they've got well developed incisors as well as flattened molars and premolars. The reason the molars and premolars are flattened is that when they chew, they're able to grind the plant material and release the nutrients from that, allowing for an easier process of digestion. However, if we compare that to the teeth in the carnivore, we can see these sharp triangular teeth that are there for crushing and biting and reducing the size of the meat that is being consumed. So distinctly different is skull B, which is clearly of the herbivore. The next question points to the carnivore, and as I mentioned, the giveaway for this individual is the large canines, and also you see these teeth which are triangular shaped, and it is typical found in a carnivore which consumes a diet rich in meat, protein, that it uses its teeth to crush and to mince bone and protein. Right, let's see, provide reasons for your answers to the above questions. As I just pointed out, I've explained the adaptations of these triangular teeth for crushing bones and the flattened teeth for grinding plant material. Guys, one last question before we have a wrap. Does skull B have carinaceal teeth? Explain your answer. Okay. No, it does not, because it's got flattened teeth. These teeth that have the triangular shape are the ones that are adapted for being a carnivorous diet. So guys, for me, that's a wrap. We've looked at revising nutrition. We've looked at various different questions, from labeling parts to unpacking the structure and function of different organs. We've looked at the ability of the body to regulate the levels of blood sugar. It's important that we reflect on all of that as we wrap up your revision. For me, you've been a fantastic audience. Trust that you've enjoyed the lesson. Cheers from my side. Have a baie lekker day. See you soon.